Wonderful.
Welcome to everybody. Thank you for joining us for the DEBRA webinar in July, 1st of July. Happy July, everybody. Um, I'm going to kick straight off and say we're very, very pleased to have with us Dr. Nick Veldhuis and all the way from Australia, from Monash University. So we appreciate you finding a way around the time difference to join us here. So Nick, you're the Australian Research Council Future Fellow at the Monash Institute of Pharmaceutical Sciences. Um, really impressive, really impressed to have you here. Your expertise in molecular pharmacology related to pain and inflammatory signalling, so very relevant to people with EB, and you've got an active EB research program. I've just been looking at the website where we've got your new EB funded or DEBRA funded EB project up on our website now. Please feel free, everyone, to go and have a look. I'm going to hand straight over to, well, very quickly, I'll just say housekeeping. This is a webinar, so it's being recorded. Please don't take any screenshots or make your own recordings. And all the way through, you can add questions into the Q&A. And at the end, we'll quiz Nick on what he has to talk about. So I'm going to stop sharing and hand over to you, Nick, for characterizing pain and itch mechanisms in a model of dominant dystrophic EB. If I can
Thank you,
find how to
Abby.
stop sharing, or can you just take over from me?
Maybe I can. Let's request and see what happens.
Let's see. Oh, it's all right. I've just lost a button underneath someone. Oh, I can't see it. Stop share. There we go. Brilliant.
Okay, let's give this a go.
I shall put my camera off and leave you to it, and interrupt only if if there are any issues.
Okay,
Thanks
thank
again.
you. Thanks, Abby. And I'd like to thank you once again for the opportunity to talk within this community. It's obviously I'm dialing in from the other side of the world, so it's always challenging for us to connect globally unless we are often doing the traveling to the other side of the world. So yeah, it's really nice to do it through this format. It's also nice to see some of the participants in the audience. Margarita, I'm assuming, is dialing in from Chile, and actually, I'll be mentioning some of Margarita's work today. So it's really nice to we're cross continent today, it seems. So, just a brief overview of where we are based. I'm at Monash University, but there is a rather large campus on the other side of town. It's about a 40-minute drive from where we are down in the southeastern suburbs. But in Melbourne, the Monash Institute of Pharmaceutical Sciences is actually based very close to the city centre, within this so-called biomedical precinct, where there are a number of different hospitals. University of Melbourne is there as well. And it's an excellent place to work for a number of reasons. We were once upon a time the Victorian School of Pharmacy. Monash took it over and it actually became a world-class biomedical research institute, and it covers a lot of different areas. I'm based in the drug discovery biology theme. We don't really have departments; we call them themes, and we have a lot of capability, which is essentially bringing discoveries all the way from medchem all the way through to understanding the biology, formulation, ADME-PK, and all the way towards clinical trials. So they call it a one-stop shop. For a lot of this type of work, and certainly there's because of that type of environment, there's a lot of biotech type activity, spin outs as well that are happening. Yeah, we're highly ranked in terms of our pharmaceutical sciences and pharmacy expertise, and this is largely due to the teaching that goes on here as well. And there's a number of notable discoveries that have happened over the years that have come from this building that you can see here. So with this audience, I didn't really want to spend too much time on discussing EB subtypes and all the different types of mutations and why it is so important to study EB. I think we're all here for that reason, and I just wanted to highlight a couple of things. If we just look very specifically at the epidermal-dermal junction and all of the different proteins and the mutations that are involved um, in genes that are encoding components that are essential for the structural and functional integrity of the epidermis and the underlying junction. And in particular, I just wanted to note that although there are different types of blisters that may form due to destabilization of different proteins, for example, in basal cells, we have mutations in keratin type 5 and 14, which can lead to this blister formation in the basal and suprabasal keratinocyte layers. And in contrast with junctional EB, we have these blisters that are forming quite specifically at the basement membrane below that basal layer due to mutations, for example, in laminins. And there's been so many excellent talks over the previous months through DEBRA UK in this particular online seminar series that have talked about a lot of this in great detail. Um, And finally, dystrophic EB, which is of particular interest to me. I'll be telling you today about our dominant dystrophic EB model. And as you can see, the blisters are forming underneath that basement membrane in the in the dermis itself due to this destabilization of collagen type 7 for dystrophic EB or a functional a loss of collagen 7 for the more severe recessive dystrophic EB. Now, although there are blisters that are forming in slightly different ways across all the different EB types, of particular interest to our research and the talk I'm going to give you today is that pain and itch are commonly reported by EB patients, independent of subtype. Now, this was actually a pain quality assessment questionnaire that was performed sort of almost, yeah, I would imagine it was about 10 years ago now by a group in the Netherlands, and I really find this a useful way to schematically or diagrammatically show the different types of pain that EB patients experience. Now, I am a basic researcher. I'm not a clinician, so although I have been fortunate enough to actually meet a number of EB patients through my connections to the Royal Children's Hospital down the road. It's it's very hard for me to understand by talking to EB patients about what types of pain they are experiencing. And often this is because EB patients, especially the EB kids that I've met, they're incredibly resilient. They don't really want to talk about their pain. So questionnaires like this are really important for us to understand the different types of pain. You can note that there are for dominant and recessive dystrophic EB, they talk about this deep itchiness. There's tenderness and aching, tingling and shooting pain, which is generally consistent with a small fiber neuropathy. And then you can see with EB simplex, although it is considered to be less painful in a number of with a number of different mutations. EB simplex mutations in general, all EB patients have daily moments of extreme discomfort, even in the absence of blisters. And this can be really mixed. It can be intermittent. They can have be experiencing sharp pain, or in more severe forms of EB simplex or recessive dystrophic EB, this may be considered stable. So there is a stable experience of pain, which is absolutely unacceptable. And so me meeting with these patients and our team meeting with these patients, it has really changed how I do my research, and I have a different reason to be really pursuing this. So I also just wanted to note, of course, that EB is a multi-system disease, and the pain is not just presented at the skin where these blisters are occurring. Of course, we know that there are many tissues and organ systems within the body that are affected. And I've just highlighted here the major systems that could also lead to the experience of pain in EB patients, certainly in the more severe forms. I think oral pain and eye pain would be obvious ones, and also the gastrointestinal tract. Now we're starting to look at that actually more specifically. My co-lab head, Daniel Poole, is actually a gastrointestinal neuroscientist, and so in collaboration with Alex Nystrom at the University of Freiburg in Germany, we're really excited to look at the GI tract in much greater detail, look at the changes in inflammation and, for example, visceral pain in the GI tract. Now, I also just wanted to note that these sorts of painful scenarios, there are a number of different painful scenarios that are addressed in the EB best practice guidelines. And I think these would be certainly obvious to clinicians and and things like dressing changes for bandages, baths, any sort of presentation of a chronic wound is going to be incredibly painful. But it's a little bit more challenging to understand that the pain that is in the gastrointestinal tract, for example. So that's something that hopefully I can talk to you about another time. But for now, for this particular talk, I will be focusing a lot more on peripheral pain and what's happening actually at the skin, which is a major source of pain. So to do that, I thought I actually might go back to a very basic illustration that I've used in a number of different lectures that I've given over the years. Now, it's a little bit simplistic, but it really proves a point about how pain processing works in our body. What you can see here is this long red line, which is representing a sensory neuron. So we have nerve fibers that extend out to our skin, for example, and then they have cell bodies that sit just outside of our spine called in the dorsal root ganglia. And then they synapse with these so-called second order neurons within the spinal cord. So what happens when under normal circumstances, this is not in an EB patient, and I'm just talking more generally about pain pathways, is forgive the cats. It was a very illustrative way of just showing that there is a message that is being sent along quite a long distance, actually. These sensory nerves are known as the longest cells in our body. You can imagine nerves that are running from your spine, for example, all the way down to your toes, could be more than a meter long. So these are incredibly long cells. They have to send these messages into the spine. And after they do so, they release neurotransmitters into the spine, which would then send messages up to the brain. And of course, we recognize this in a number of different ways, that there was a painful experience. So we actually withdraw or avoid that painful stimulus in the future. And of course, there's an emotional response that comes with that as well. Now, this might be slightly different in patients that are experiencing EB. It may be that a non-painful stimulus may also be perceived as pain, and that's generally called or described as allodynia. So if we look at this stimulus intensity profile over to your right, you can see here that we have a normal pain threshold here relative to pain intensity in the y-axis. And if we actually experience pain, even when we're not meant to, what you find is that there are a number of different things that happen. We get more signals when we shouldn't. Even something as simple as changing a t-shirt or changing clothes could become painful when it shouldn't, because you're feeling it in this range here. Your nerves are activated when they shouldn't be in an otherwise healthy person. And of course, not only that, because these nerves are firing because they're so damaged or there is inflammation occurring in the skin, for example, you actually can feel a heightened sense of pain. We call this hyperalgesia, or otherwise, I might describe it as pain hypersensitivity within this particular presentation. Now, to sense our environment, there are a number of different nerves that are involved in this as well. And I'm not going to give you too much of a long lecture about pain and how it all works, other than to say that something that we're particularly interested in is these nerves that innervate the epidermis, these so-called intraepidermal nerve fibers. And these specifically described as non-myelinated nociceptive neurons. For example, there's a subtype of neurons known as C fibers, and they are particularly useful or important in our body for sensing thermal and mechanical pain. And there's also non-myelinated A delta fibers. And together, these fibers that are innervating the skin, they are required for sensing pain. And of course, we've got a number of other structures in the skin as well that aren't necessarily there to sense pain, but other types of stimuli. So to allow us to sense our environment. For example, we've got Merkel discs, which allow us to perceive shape and texture. So that's a very mechanical sensation, but it's got very little to do with pain unless there is a considerable amount of force. So I mentioned Margarita Calvo at the beginning, and this is some excellent work that she did with David Bennett. This was published back in 2017, and I wanted to just show this as some of the early work that's been done, just to try and understand how nerves might be different in EB skin, EB patient skin. So what we're looking at here is the some of the work where they were staining. Nerve fibers from EB patients. And if you look in the most severe cases, such as recessive dystrophic EB, you can see that there is a peripheral nerve fiber loss. And this is not only in the epidermis, that's this upper region you can see here. The junction is labeled here with this red dashed line, but it's also apparent in, for example, sympathetic nerve fibers that would be surrounding sweat glands, for example, because it's also noted that EB patients have differences in the capacity to sweat. So there is a very strong nerve difference that is occurring within EB skin, and it's very easy to say that the skin is ultimately being changed. There's a lot of scarring and wounding that is occurring, certainly in more severe forms, such that nerves can't actually make their way into the epidermis. But there are a lot of other things going on as well. And so that's where our work comes in. We are really hoping to look at less severe forms of EB, such as dominant dystrophic EB, to understand if this also occurs, because very little is known in less severe forms by comparison to a lot of the work that's been done in recessive dystrophic EB. And try to understand a lot about the molecules and cells that might be actually changing how nerves innervate our skin and enable us to sense our environment, and of course, how that might actually change how they fire and send messages to the spinal cord and up to our brain to perceive pain. Now, look, this is following on this slide here from Matthew Cayley. He actually presented some excellent slides a couple of months ago, really talking about how challenging it is to bring any drug to the clinic. And I just wanted to highlight that this is actually even more challenging, I would argue, for analgesics. Look, even though in the population it's hard to put numbers around this, but they estimate that at least 1 in 5 suffer from chronic pain during their lives. And I'm not talking about EB patients here, just any type of pain. And there are many, and obviously presents in many different forms. So you can see, even though it is incredibly prominent, and we know, for example, that there are major challenges with opioid use, and there is a big movement away from opioids within the clinic. And despite the fact that chronic pain is a massive cost in societies around the world, in developing countries, it's in the hundreds of billions annually in terms of lost productivity. Despite all of this, the discovery path is relatively low. Pharmaceutical industry doesn't really want to invest in analgesics the same way that they may want to for other conditions, other diseases. And there's a number of reasons for this. In particular, we know that there's a massive attrition rate once you move from preclinical studies all the way through phase 1, 2, and 3, for example, in the market entry. And this is for a number of reasons. Obviously, you've got to pass through healthy patients, make sure drugs are safe, and then you need to show that they actually work. And for pain, you really have to shift the needle a little bit to actually provide convincing data to suggest that you might have something that might outperform current analgesics that are on the market. And in addition to that, there's a magnifying glass on this type of research. They're looking very carefully to make sure that analgesics, for example, are not addictive, so that we don't repeat the opioid crisis that we're currently faced with. And so you can imagine that this makes it incredibly challenging, and even more important for us to be for people to be looking at pain for these rare diseases, diseases with orphan designation. Now, there are limitations to drug development, and I won't touch on this in a great amount of detail, other than to say that that major challenges include that we really need safe drugs. We're not very good at predicting adverse side effects. We don't always have good preclinical models. It's also very challenging to get drugs to target affected tissue and not other tissues within the body, and also to make sure that drugs aren't metabolized too quickly, for example. And to address these limitations, there are some really exciting advancements. And so it is a really exciting time to be doing this type of drug discovery research in the pain space. We now have better preclinical models, and I mentioned Matthew Cayley and Emmanuel Ragnoni's work, for example, in developing a one-stop shop, like a service, so that people can actually have access to a range of different preclinical models. I think that's a really exciting advance for the EB community in particular. There's this new omics era, and there is capacity now to look at molecular and cellular pathways across many different cell types simultaneously. There's almost too much data, and so there's a lot more power in AI-powered drug discovery and analysis now as well to actually handle this as well. So there are a number of things that are allowing us to really make some key strides in this space. And one of the ways that we're doing this is by developing and looking more carefully at a milder form of dominant dystrophic EB. And so I just wanted to briefly mention the mice that were developed by Ken Pang at the Royal Children's Hospital down the road. So we use CRISPR to actually generate a number of different dominant dystrophic EB mutations. And taking this glycine to arginine substitution, that's position 2043, which is the most common form of DDEB in patients. They characterized these mice quite carefully and showed that there were actually blisters around the mouth in suckling pups. That actually resolves after about a week. But then you can see, if you look carefully at the paws, that a number of different micro blisters will occur across the paws over approximately a 3-month period. And then you can also see, in addition to the blisters, nail loss, and all of this phenocopies dominant dystrophic EB patients. So this work was published back in 2021. And around that time, so we're just talking in the middle of COVID, Ken Pang came to me and actually asked whether I'd be interested in assessing whether these mice also experience pain. So the way that we do this, there's a number of different behavioral studies that we perform. I think I might have a video later on, but we can actually, for example, look at their mechanical sensitivity. For example, we can put a mouse on a mesh stage like this, and then we just gently touch these filaments into their paw. They're not normally painful. And however, if you have a mouse that, for example, has some discomfort and sensitivity to this stimulus, they'll actually withdraw their paw. So this paw withdrawal, a rapid paw withdrawal, is given a low score. So this is not an exact measurement of pain, but it is a representation of a mechanical pain hypersensitivity. So you can see that these mice actually showed quite a clear difference relative to their wild type litter mates. We can also use an infrared light and shine a beam of infrared light onto the paw of the animal, and that will be a subtle warm sensation, and that would they will also withdraw their paw to that. So we can see that there is a mechanical and thermal hypersensitivity in these mice. And that was actually really exciting because it allowed us to actually move forward and start to test these mice more thoroughly in the hope that we could try to dissect the mechanisms involved. And so just to look in particular at this paw withdrawal, using these von Frey filaments, so this mechanical hypersensitivity, we started to look more carefully over time. You can see up to 6 months of age that you can see this sensitivity is maximal at around 12 to 14 weeks of age, and then it seems to resolve over time. We don't know the exact reason for that at this point. We'd be open to any suggestions, but we predict that there's probably a greater level of fibrosis in the paws at these later stages, and that might be consistent with prior preclinical pain studies and recessive dystrophic EB mice, where they see a loss of mechanical sensitivity, potentially due to scarring, actually preventing any feeling of those filaments being advanced into the paw. And we can see that there's no difference between male and female mice, which was also really important. Then we actually wanted to assess whether these mice would be really useful for analgesic testing. That's really critical for us because we may, we hope to identify pain targets, but we also want to know that if we actually, for example, introduced or tested a range of different medicines in these mice that might block or reverse pain, that we may actually observe a change in their behavior. So we consulted with the Royal Children's Hospital palliative care team down the road, and they actually indicated that pain management in EB kids, for example, is quite ad hoc, and there's quite a long list of different analgesics that they may use at any given time. So what we decided to do, and I will note that this work is actually unpublished. We're in the middle of preparing a manuscript, so you're actually seeing it hot off the press before it gets published. We actually took analgesics from all the major classes, for example, and one opioid, one non-steroidal anti-inflammatory agent, one anti-convulsant. For example, we also looked at a tricyclic antidepressant, which is also frequently used for chronic pain, probably not so much for EB patients. We looked at anxiolytics and also a range of other different candidates that hopefully you can see here, and the resolution holds up on the video. And what you can see, what we're looking for here is a higher scores, poor withdrawal scores. So you can see with different doses, for example, of buprenorphine, which is a very well-known analgesic, that as we dosed higher, you can see that they weren't as sensitive to these filaments being advanced into their pores. So they're not withdrawing them as easily or as readily. Gabapentin, a very well-known neuropathic agent, which has actually been more recently tested in the clinic for EB patients, was also shown to be analgesic in our mice, as was meloxicam. And in fact, that was probably the most analgesic response that we saw, which suggested there was a strong inflammatory component to the pain that these or the nociception which that these mice were experiencing. We've also, I will just very briefly touch on some of the work we've been doing with Professor Ian McGregor and John Arnold up at the Lambert Initiative for Cannabinoid Therapeutics in Sydney. We know anecdotally that cannabinoids are used in EB patients and in adults, for example, for pain relief and anti-inflammatory purposes as well. And so what we've done is we've focused on non-psychoactive plant-based cannabinoids. And although, I think the obvious gains would be with topical administration of said cannabinoids, our early work has been with intraperitoneal injection into these mice. So you're talking about essentially a preclinical administration. This is not a typical route of administration that is used in patients, but it's a very effective way to quickly assess whether certain drug candidates may be working in your mouse model. And just taking the example of CBD here, you can actually see that we did see some analgesia in all mice. And if we looked more carefully, we actually see, unlike the prior studies that I just showed you, that there was actually a sex-specific difference here. And we believe this is probably due to the route of administration that we used, and that we probably need to look more carefully using, for example, intraplantar or topical administration to see if that holds up, or if we can actually see pain relief in a non-biased way in these mice to really pave the way or open the door for us to do a lot more cannabinoid research to assess efficacy and safety of a range of different cannabinoids. And we're really excited to continue this work, and it's definitely ongoing with support from Ian and John, and the Lambert actually providing some financial support. Now, very briefly, these mice, we've also assessed itch, and we actually can't see any major difference if we just look at their scratching behavior. In this particular case, what we're doing is we're counting the number of times that they advance their hind paw to their neck and scratch. This is work in progress. We're actually going to look more carefully at their biting and licking type behaviors as well to see if there's anything different there. We know that, for example, junctional EB, there's a strong itch phenotype there. Dominant dystrophic EB, that might not be so much the case. And certainly, even if we actually try to explore this further by, this is just group data for what I showed you in the previous slide. I might just sort of slightly jump ahead to show that baseline itch, if we just look at the total number of scratching bouts, For dominant dystrophic EB mice, we consider there's no major difference if we're looking at around 3 months of age where we saw the pain phenotype occurring. We see no difference in male and female. And even if we administer an itch-inducing peritogen into the nape of the neck of these mice, that they weren't actually scratching anymore. So it didn't appear, at least with training a camera above the mice, that we can see any major differences. But we're going to explore this further because we do believe this is a really important characterization that or data package that we need to put out into the community and publish, hopefully sometime in the next 6 months. Now, just in the final part of my talk, I really wanted to discuss what we are doing here to explore what we're doing at Monash Institute of Pharmaceutical Sciences to explore and dissect the cellular and molecular pathways which might be leading to this pain behavior that we've seen in these mice. And so the pathophysiology of pain, look, I say it's unknown. I think we know that there are some pretty clear reasons why EB patients are experiencing pain. But it is incredibly complex because we have, for example, direct damage to nerves. We know there's an excessive amount of inflammation in the dermis, for example. Fibroblasts and immune cells could all be communicating to nerves to actually warn the skin that there is and the nerves in the skin that there is a danger signal there. And so we wanted to, I just wanted to touch on a few things about how you look at this. And I liken it to looking for a needle in a haystack. Actually, Abby, I do have a few more minutes, I hope. Is that okay? Yeah. Just before I go on. Yeah, we're good. So, look, a lot of analgesic or target discovery work you could liken to a needle in a haystack. And look, there are a number of different ways that you can essentially try and identify or find the so-called needle within such a complex multifactorial disease. And essentially, the way that I look at it is, if you need to find a target, you essentially need to throw the kitchen sink at it. There are so many different ways that you need to look at samples using a range of different complementary, multidisciplinary techniques to try and understand what's going on. And we are doing this in mice. And of course, once we have a much greater understanding of what's going on in the skin of these mice, we hope to bring this over to patient samples. So this is our kitchen sink. We use high resolution microscopy. We use a range of different omics techniques. For example, proteins. We look at all the different proteins that might be changing. We can do this in whole pore proteomics. We can take pore pads and look at all protein changes in the skin. We can also look at the secretome using, for example, keratinocyte or fibroblast 2D or 3D cultures to see what they're releasing, how this might actually change the function of nerves in different ways. We're also using another different type of omics technique, lipidomics and metabolomics, which is spatial. So this is a mass spectrometry-based technique where we can spatially profile small proteins, peptides, lipids, and other metabolites that might be changing across skin sections. And of course, there's a lot of functional validation that follows. For example, characterizing sensory neurons by calcium signaling electrophysiology, for example, and testing various analgesics the way that I've shown you in the previous slides with our behavioral studies. And so we're looking mostly at the pore, but we're also looking at these dorsal root ganglia, the cell bodies of these sensory neurons, and also into the spine to see what has changed across all of these key sites within the pain processing pathway. And so this is our EB pain research network that we have in Australia. I just wanted to note Dan Poole, my co-lab head, Johannes Kern in particular, who's the clinician who's been heavily involved in this work, and also Wendy Imlach, who's done a lot of the behavioral work that I've already showed you in this talk today. And Danya was heavily involved. She's a PhD student. There's a photo of her. We were all at her wedding, and she did a lot of the behavioral work here. And also noting our work with Alex Nystrom in this particular program, who's based at the University of Freiburg in Germany. So just a whistle-stop tour of some of the things we've seen in our mice since we started this characterization. looking at PGP, as for example, I mentioned Margarita Calvo and David Bennett had done previously, we can actually see that yes, there are nerves in the epidermis of these mice, but it seems that these nerves are actually changed. And we're actually seeing the loss of particular nerve fiber subtypes. CGRP is a peptide that's known to be present in a certain pain transmitting or nociceptive neuronal nerve fiber subtype within the epidermis. And we're seeing a loss of these within the epidermis, which is suggesting to us that there are signals there within the keratinocytes which are telling nerves to actually not innervate this particular damaged tissue. Or it could be that there are structural changes to the keratinocytes themselves, which actually prevents these nerve fibers from actually innervating the epidermis. We've also, with the proteomics, we've seen some really exciting things. I'd be happy to talk to anyone about this at the conclusion of this talk. And what we've done here is a comparison of male and female EB mice to wild type male and female. We can see, in fact, if you look at males and females separately, there are incredible differences in the proteins within the skin, not only of EB mice to wild type, but also just within the wild type cohort or the EB cohort. And so this is just a heat map just showing that there are many different proteins. We saw 3,000 significantly changed proteins within a whole paw proteomics analysis. We've seen around about 10,000 proteins in total, which is an incredible number to be seeing with this new mass spec instrument that we have available to us within MIPS. And just looking under the hood, looking more carefully at some of the data, I just wanted to note that we actually do see this reduction in COL7 within our mice with this mutation. And so that was actually a relief when we saw this. But we also saw a range of different collagen differences, both within male versus female wild type and also from wild type to EB. I just wanted to note one in particular, for example, collagen 17. You can see there is a reduction both in male and female EB mice. So, for example, just this single amino acid change within collagen 7 is having this knock-on effect and influencing a number of other collagens within the skin. Now, we can also use a lot of bioinformatics to filter down and try and understand what is going on exactly within the skin and how these protein changes may be influencing pain pathways. And essentially what we can do is, for example, analyze paw skin or secretome from skin cultures. We can use transcriptional workflows. So this is workflows that are typically used for analysis of transcriptomics, and we can use it for our proteomics pipelines to actually filter for secreted proteins that are likely to bind sensory neurons and then determine whether they are druggable targets or not. And then this enables us to go, for example, from 3,000 significant changes down to very few. So at this point, what we've done is triage, for example, from 9,400 proteins all the way down to 34 significant pairs of proteins that are secreted that may be acting on sensory neurons that we are now moving into behavioral studies to validate these. So it is a very exciting prospect. And through DEBRA UK funding, we've been able to actually explore this over the next couple of years in much greater detail. So we're very excited for that support. And finally, we can validate this with spatial imaging as well, as I mentioned. So this is a relatively new technique where you can actually take skin sections. You can immunostain some, you can take others, and you can actually shoot them with a MALDI-TOF laser. So it can actually zap samples down to 5 micron resolution, so about to single cell resolution. And in every single spot, you will get a complete metabolome lipidome profile across skin. So just to give you perhaps one example of what that looks like, just looking at cholesterol here, you can see there's an abundance here. And this is actually sitting at the dermal epidermal junction. And then in our mouse, our dominant dystrophic EB paws, you can see there's actually a loss of cholesterol. This is consistent with other work that's been done in junctional EB, for example, with Matthew Cayley and colleagues. They've actually put out a bioarchive preprint, which indicates something similar. And so what I'm basically trying to highlight here is that we have bulk, like immense, huge data sets of proteomics that are showing massive changes, for example, in lipid metabolic processes. We can see that these are down in male and female. And now we can actually start to spatially see that these are also occurring within pore skin. And we're really excited to carry on this work and actually look in EB patients as well to profile lipids and metabolites more extensively. So just to summarize, our mouse model, we believe phenocopies the dominant dystrophic EB skin fragility and pain that is experienced in patients. And we propose that it is a novel skin pain model that is going to be incredibly valuable for analgesic discovery. And we've got a lot of exciting ways that we can tackle this problem. And the early insights from our analgesic discovery process is that meloxicam, an anti-inflammatory agent, and cannabinoids are showing some effect, and it does suggest that there is a strong inflammatory component, which is very well known for EB. We see that there is a disruption in nerves. It's not the same in the dominant dystrophic EB as what has been reported previously for the recessive dystrophic EB. And we are starting to dissect this apart further to look at, for example, axonal guidance or nerve guidance cues that might be changed within the skin of these mice. We see very clear sex-specific differences, and we need to be mindful of these when we're doing carrying out this analgesic discovery process in the future. And I also just wanted to note that we believe that there's probably a change in the pathogen load, for example, different colonization of bacteria in these skin, which is also influencing our proteomics data sets so far. And I think this is also going to be really important for determining how pain chronification occurs in EB as well. And I showed you photos before, so I don't really need to do any other acknowledgements other than just our funding from the NHMRC, a lot from DEBRA Australia over the years, DEBRA Austria previously, and now with DEBRA UK. So we're really excited to continue this work. Thank you.
Thank you. It's absolutely fascinating. I know there's no questions in the Q&A yet, but I'm bursting a bit because there's such a lot of there's such a lot of information that is coming out of the multiomics stuff once we have a mouse model that does do this that we can use to study pain. I think it's something that I was a bit skeptical about. I was at a meeting and so somebody had a poster on pain and using mice. They were saying you can tell by the mouse's face whether it's experiencing some pain. And
Yeah.
I was really, really, but this is they were convincing, and this is very convincing that you are able to show that the analgesics are working. Um, Gosh, yeah. So the sex differences to start with. I mean, I know some people might be going, oh, of course, men and women feel pain differently. Um, I'm just relating that back to what you said earlier about the sort of ad hoc treatment of children with for their EB pain. Do you think is there too much difference between people for all their sort of human factors, all our genetic differences as well as sex, all the differences that are there? Does that make it harder to pinpoint what works and what doesn't? Or do we just have to address this very carefully with the knowledge that that is the case?
Yeah, I think there's two sort of sides to this answer. I mean, I think, look, it is true that there may be, specific medicines that are more effective for males or females in different pain contexts. Like, for example, for the same or a similar pain experience, we know at the cellular level there's different types of signaling pathways that are involved, for example, higher up in the spinal cord and also locally, for example, in the skin. And this is the contribution, for example, of immune cells or immune-like cells, T cells are a really obvious one, and then microglia in the spine. They're quite different between male and female. So it is possible that we may need specific or bespoke medicines, but I don't think you could convince the pharma industry that this would actually be a useful way to tackle or the development of any medicine. What you really need is something that would be equivalently valuable to both patients, and then of course, there's a conversion of them financially for that. But yeah, so I guess having said that, when we are looking at targets, we need to be mindful that there might be slight differences and that we need to really look at targets that might be useful in all pain scenarios. Yeah, so Um, Look, and the only other thing to add about the cannabinoid work is I do really believe that that is an administration route risk limitation, and so I don't think we would see the same thing, for example, with a topical cannabinoid, and we're
Right.
really excited to try
I
that.
mean, this was going to be my next question, really, because we did fund a small study on cannabinoid oil. Um,
Yeah,
in the Netherlands, people were using that alongside their regular pain relief. That finished a year or two ago. We've had the results have been published, and they weren't massively exciting. We weren't able to rush out and say to everyone, "This is it. This is going to really help." But I think it was quite a
yeah.
small study,
Mm.
and if there was a sex difference, I don't know whether that would compound. You'd need more people, presumably, to pick apart what you're seeing. Um,
I think so. Yeah.
but there's so much sort of anecdotal and scientific evidence that cannabis can help people. I think that it's worth staying on top of. So yeah, I'm just thinking of I must go back and have a look at the previous project. And Nick Schrader was the guy who was doing most of the work on that. Marieke Bolling,
Yeah.
yeah.
Yep. Yep. And
Um,
I was showing questionnaires from him today. Yeah. So that
oh right, yeah,
yeah, it's really yeah. I'm aware of that study, and maybe just a few other comments there. And I think coming from an institute which is actually really well known for its drug formulation, drug delivery expertise, that I think that is also crucial. So it is easy to say if you've got an oil that you would hope that it would absorb the skin.
yeah.
But I think there are gains to be made by using very specialized formulations, which may actually allow penetration of different pharmaceutical agents, not just cannabinoids, in different ways. So formulation can be critical, for example, for the duration, like the dwell time of a drug, for example, in the skin before it gets cleared, how deeply it penetrates. And so to say that oil might be, showing some pain relief, it doesn't mean that there won't be other formulations that might show something different as well.
I think this is absolutely key, how we get stuff to the skin. So this, I think, the study we did was
Yeah.
oral cannabinoid oil, but yeah, getting stuff to where it needs to be, getting the treatment where it needs to be. There's a couple of questions, or a comment saying that the problem with cannabis is the risk of psychosis in the young patients. And I was going to ask about the differences with children and adults, but a question from Margarita. What do you think about itch? Is it neuropathic that people are feeling with EB?
Do you mind if I answer the cannabinoid one first, just so we move
You'll stay on the
while we're talking about it?
cannabis. Yeah, sure.
Hope Margarita doesn't mind. Thanks for your question, Margarita. So look, I think in terms of psychosis for cannabinoid, that it is quite specific to the full agonist THC for CB1 and CB2 receptors. We're looking really carefully at this. There is an argument, for example, for using THC in other settings, and it might not have the same psychoactive effects if it's not taken orally, for example, or intravenously. And so, yeah, I don't know the answer, but all I am saying is that it is different, and it would be worth looking at a different settings. Having said that, I think the appetite, like the risk, is quite substantial for looking at something that might have any type of psychosis effects. Certainly, when you're looking at medicines for children. Now, that's the other reason why we focus on phytocannabinoids at this point that are non-psychoactive substances, of which there are many. We've been testing all the obvious ones: CBC, CBD, CBN, CBG, and we actually think there are a lot of gains to be made there, and also in combination as well. And they're relatively safe, so you can actually use them in pretty high doses locally, and we hope that that might actually give us some insight. Secondly, about itch, it's a really interesting one. Is it neuropathic? I think yes, but I think there is a combination of just fibrosis and scarring that might also add another layer to the itch processing that's occurring. For others that aren't quite as familiar, and I would argue I'm not as familiar as Margarita as well, so apologies, Margarita, that I probably don't give a complete answer to you, but um, something that can occur is if your neurons are fundamentally changed in any disease, you can also get changes in the neurocircuitry and the processing higher up, for example, in the spinal cord. So, for example, you can get confused signals, things firing in different ways, and these can be quite, although they're described as plastic, they can be quite permanent with persistent damage, for example, in the skin. So I do believe, yes, that it is neuropathic, and that comes back to this type of deep, itchy pain that I think that for example, RDEB patients are frequently describing in questionnaires.
Thank you. Yeah, I mean, yes, the hope is there that there'll be something that is very targeted for pain relief, but without the drowsiness or the addiction. And I know people wonder how well mice mirror. I'm like myself, wondering really how well mice we can see how much mice are in pain. Presumably, mice are l- shorter lived than humans. Are there standard protocols for measuring addiction? Can you see mice becoming addicted? And presumably, you can see
Yeah.
them becoming drowsy. These common side
Yeah.
effects, are they?
Yeah,
Yeah.
absolutely. Yeah, no, you really can. So the drowsiness is a really easy one. I think, for example, if I gave a mouse certain doses of opioids or THC, for example, THC is probably an obvious one. You can literally see mice look stoned, like they're sitting in a cage,
Right.
and they don't really respond to these filaments because they can't be bothered, so to speak. And similarly, there's an apparatus called a rotor rod where you can actually allow mice to there's a rotating rod and they will actually maintain themselves on this spinning rod, this slowly spinning rod for a period of time if there's no psychoactive effects. However, with THC or opioids, for example, you would see that they don't last as long and they would drop off the rod into the bottom of the cage. So you can test drowsiness very easily. In terms of addiction, there's a lot of work in that space, across the globe and including in our institute. There are all sorts of amazing ways that you can assess that in mice. For example, in our institute, they're looking at addiction and psychosis, for example, and using touchscreens. So you can train mice to use touchscreens, and they will might get, for example, the strawberry milk hit every time that they touch, identify a certain shape, and then you can actually then start to look at their returning behaviors in certain settings with different pharmaceutical treatments. So there are a lot of amazing ways that you can very deeply analyze mice behavior. And I think we are starting to understand more about mice behavior in the way. And I did use the word pain loosely for mice for this talk. We really still do not call it pain in mice. It is quite strictly described as nociception in the absence of this emotional response. But yeah, even now with facial grimace scores, you can see there's a lot more going on, and there are a number of other behavioral traits as well that you can look at now. That we're excited to do in our mice as well. Actually, we're writing ethics for that at the moment.
I think, yes, ethics is tricky. I know we have some of our member reviewers who always comment that they're uneasy about the use of animals, but I think the way you've described it, we can see the value, the absolute value of using mice in this way. I'm just wondering, in that context, can you, the fear of pain and the anticipation of pain, those functions of pain that allow us to protect ourselves from repeated harm, do you think this is something also that you can see in mice, or do they not have that level of sort of cognitive functioning?
Mice are incredibly tough creatures. They really are brave. They really don't want to show pain in different settings. For example, even in the presence of male researchers versus females, they'll actually behave differently. So they're very sensitive to pheromones, the types of perfumes that people are wearing, and things like that. Whether they can hear other mice in pain in cages in the same room, that can change their pain behavior. There are many different ways that they are influenced, and in general, yeah, they're otherwise. This is a survival mechanism for them. So yeah,
For us all, I guess.
it's
But
very complex. Yeah.
okay, so they are a good model. They're a good model. It sounds like this is a very good model to have.
It absolutely is. I think that, yeah, you just have to tread carefully. We do a lot of work with our ethics committee to make sure that we're doing this in an appropriate way, and these mice are incredibly well looked after to ensure that they otherwise are moving freely around cages. They're otherwise quite happy, exhibiting all other normal mice behaviors, unless we sort of challenge them with these very mild, um, thermal or mechanical evoked
Yeah.
stimuli. Mm.
Thank you for covering that because I think it is relevant. It is relevant for people, and I know that's one of the issues with modelling EB is that some of the forms are so extreme that yeah, mice may not survive those. Well, yeah, they could be very life limiting. Um, I can't see any more questions that have come in, and I think it would be a good time to stop, give people just a few minutes back on their hour. But we really, really appreciate you having taken the time to come and speak to us about this. Looking forward to the results of
Pleasure.
the project. And yeah, absolutely brilliant. Thank you to everyone for attending. If you do have any questions that crop up, you can always come back to us. And please do leave a bit of feedback or check out our website where we've got information on those projects. So I'll say thank you again, Nick Veldhuis. See you
Thank
again
you.
soon, hopefully. Thank you very
Okay.
much.
Yep.
Goodbye, everybody.
See everyone.
Bye.